Achso, können wir anfangen? Das ist wie im Fernsehen hier. Oder darf ich nichts mehr sagen? Okay. Yeah, hello everybody. It's my honor and pleasure to welcome you to the Anneliese Niethammer Lecture organized by our Collaborative Research Center 1313, and is a very special atmosphere here. It's the so-called Kuppelsaal, and normally you can see the stars, but today you will see only one star. And it's my honor and pleasure to welcome Professor Ingrid Kögel-Knabner. We know us very well, 20 years ago. She was the boss of a team from the DFG, I2, and we have had meetings together to discuss what's going on for the future. But let me come to Ingrid Kögel-Knabner. She has studied in Bavaria, in Bayreuth, geoecology, and after the diploma, she did a PhD, and she has finished her habilitation in 1992. And then she did a very good decision. She went from Bavaria to my home area, Westphalia, to Bochum. But that was not really a long trip. It was a long trip, but she went back in 1995 to Munich, to Bavaria, to the TUM, to the Technical University of Munich, and she is the head of the Institute for Soil Science. And she is well known, not only here in Germany. She is one of the soil scientists worldwide. And I will give you three impressive information about this. She is the member of the Leopoldina and the Accatech, the Bavarian Academy of Science. Then she got the, I think it's the highest ranked award in Europe for environmental studies last year. And that was a handshake with our president, correct? And that is really impressive. And today she will give a presentation of mineral surfaces and matter accumulated in soils. I think it's not so easy for me to have a look to the roof here. Ingrid, the floor is yours.
Thank you very much. Thank you for inviting me to Stuttgart. It's a pleasure for me to be here, especially in these specific surroundings. It's great. And yeah, I hope you enjoy with me some minutes on mineral surfaces and organic matter accumulation in soils. Like always, I did not very much of this work. I would like to acknowledge collaborators, Carmen Höschen and Steffen Schweitzer, who are still with us in Technical University of Munich, but also Angelika Kölbel, now in Halle, Carsten Müller, who recently took over a professorship in Copenhagen, Michael Steffens, who is in Switzerland, and we collaborated also with people from DLR in Oberpfaffenhofen, Derek Rogge, he now went to Canada, and we also collaborated with Steffen Schlüter from the soil physics in Halle at UFZ. And I would like to acknowledge all their contributions here before I now go into details with why do we study organic matter here in soils? Because soils are a large terrestrial carbon pool, and they are part of the terrestrial carbon cycle. Organic matter comes in via CO2 from the atmosphere that is assimilated via photosynthesis in the vegetation, and these can be forests or shrublands or, of course, croplands. And all the above ground litter and the crop residues enter the soil also as litter materials. Roots die and decompose in soils, and we have, this is become very obvious in recent years, a large part of the organic matter that comes into soils comes from rhizodeposition. And all of these materials are decayed, decomposed by microbial activity, heterotroph decomposition, and most of the carbon is mineralized again and goes back as CO2 to the atmosphere. But a small part remains and forms soil organic matter. And as this occurs year after year, we have a very large terrestrial carbon pool. It's much larger than the living biomass and several times also larger than the carbon pool in the atmosphere. And this also means whenever we change or manipulate things in soils, we change this accumulation of organic matter, and we can also manage the accumulation and storage of carbon in this pool. And of course, this means if we can store a lot of the CO2 via this cycle, we can also take CO2 out of the atmosphere, because the soil organic matter that is in the soils is stored for quite long times, and we have fractions that are hundreds and thousands of years old in different soils. This is some German soils, and this is the amount of carbon, organic carbon in tons per hectare, and you see soils can be very different and store in the topsoil a lot of carbon, but also when we look closer and when we analyze the soils even in the subsoil, where we do not see so much organic matter accumulation, there is a lot of organic matter accumulation, and as these are large volumes, the subsoil contributes large carbon storage and also a large carbon storage potential to soil in a global aspect. So how does this happen? Mainly, as I've explained, we have the root and leaf litter that is decomposed, and we have the root exudates, and all of this is decomposed by the microbes. This is a gimmick of microbes, and the microbes eat themselves and also produce microbial residues, microbial necromass, and when they decompose the plant materials, they also produce soluble organic matter. It's a small proportion, but quite relevant in some soils. And the plant residues that we have here become more and more decomposed with time. And all of these materials and what the microbes produce, so-called microbial extracellular polymeric substances, are also decomposed and exudated, and they all interact with the minerals that we have in soils. And these are basically clay minerals. They have a specific shape. They are, have a platy shape, which is, I think, finally quite important. But we also have, and this is just a sketch also, oxides, mainly iron oxides, in many soils. And we can have sorption of dissolved material. These residues accumulate at mineral surfaces. Basically, the surfaces of the microbes are very attractive for minerals, and together with their polymeric extracellular materials that they exude, they attach these materials and themselves to the mineral surfaces. And on a larger scale, also the plant residues, when they decompose, attach a lot of mineral material. The minerals are here really these small black platelets, and they form larger aggregated structures. It is important that this all goes basically, and of course the microbes release CO2, but not all what they eat is released. Some goes into this part that is described here, and this is what we are interested in. So it is important that these heterotrophic microbes have a suitable habitat in the soil so that they can fulfill these functions. If they do that, of course, we see that here we see again the iron oxides, the clay minerals, these organic materials, and they attach together to larger particles. This is difficult to put in one figure, and I'm not sure if I really was able to do that, but we have then larger aggregates. Some of them have plant residues inside, some of them have roots attached, and these aggregates are then in the sand size, which means 2 millimeters, and they are built of these smaller microaggregate units that are smaller than 20 micron, and these building units are smaller than 2 micron. So we have a number of scales that we bridge in soils, but most important is that if a soil is well fed with organic material, we get a pretty porous structure that is stable for many, many things that we can do with soils. So we can not only walk on them, we can also drive on them. We can drive with very heavy machinery on them. They are resistant then to erosion by water or wind and have a stable structure. So generally, this soil architecture then defines a very complex and heterogeneous interface in soils, because these minerals all and the organic matter have exchange capacity. They are able to exchange cations, anions. At the same time, we find very nearby, and this means at nanometer scale, hydrophobic surfaces. And of course, I guess most of you know that better than I do, they produce a porous medium that is filled with air and water. It can swell and shrink. It controls the storage of water in landscape. It controls the transport of water and solutes. And at the same time, it is a habitat for microorganisms and a kind of a self-organized system where the microbes live in, but also where they produce their own structure. And this also means that most of the organic matter in mineral soils is bound in these aggregates. And this aggregation and the soil's carbon status is maintained by continuous feeding of the soil heterotrophic community with the fixed carbon from, as I said, above ground and below ground litter, crop residues, root exudates, whatever we add to the soil or nature adds to the soil helps to store organic bound carbon in the soil. But it also means that the type of land use and the soil management that we do is decisive for carbon storage. And so what we are interested in is how these processes work at the submicron scale and how this has a feedback to the carbon storage potential and the carbon storage capacity of a soil. So what I would like to go into more detail is how mineral surfaces and organic matter accretion work in soils, what the spatial distribution of organic matter is at the microscale, and how this structure formation, this aggregate formation works as it determines a lot of the soil functionality and, of course, the porous system. The approach that we use very often is we look at soils in the field, so we have a lot of field investigations. We combine it with targeted and, we think, well-designed lab incubation experiments, and this is combined with high-resolution analytics. This is our NanoSIMS. NanoSIMS is secondary ion mass spectrometry at the nanoscale, and I will explain shortly because I will show a lot of such NanoSIMS data. What you do is you hit a surface, and the surface has to be very, very flat with primary ions. In our case, it's most of the time cesium, and you sputter off, when you hit the sample surface, you sputter off so-called secondary ions. These secondary ions are then separated according to mass, and what we finally get if we do this very often in a scale of, this is, I think it's difficult to see, this is 5 microns, so totally it's 25 to 25, a lot of what I will show, or 30 to 30 micrometers. And what we can get is at the same time a distribution of elements, but also of isotopes, so we can take advantage of the fact that we can separate the C12 mass from the C13 mass and can use labeled materials that we add to the soil and that decompose in the soil, and we can trace the organic matter that comes in. And I will show an example for that. We very often use 16O because 16O gives a nice surface of the minerals in general. Nitrogen, which is an important component of the organic matter, comes as CN, so we can look at 12C14N, we can also look at 12C15N, also a nitrogen isotopic label can be differentiated. And when we want to know more about the surfaces and their mineral composition, we can look at aluminum or iron. And we can, if we have samples that are really flat enough and we take a lot of other precautions, then we can go down to a lateral resolution of 50 to 100 nanometers. 50 nanometers is basically what the machine is able to do, but yeah, we are often more in a lateral resolution of 100 nanometers with the cesium source because the sample is not perfect. And we sputter off probably at least 1 nanometer, but that depends a bit on the type of the material that we hit with the cesium. So I would like to go with you then in one of these lab incubation experiments that we did, and we looked how organic matter associated with the fine fraction is newly formed, and we used a unlabeled litter material to the soil because we always want to control, and we used a label that we added as in the plant residues. So we had labeled plant residues. I think in this case it was potato leaves that have a 15N and 13C label, and we incubated them for 42 days at controlled conditions. And what we found was when we added this to a soil, a topsoil, an arable topsoil of a farm in Bavaria, north of Freising in Cheyenne, and such a Luvisol AP horizon is a typical arable topsoil in Bavaria and generally in Germany. So what we find, this is then with time the label of 13C and of 15N, and we see that immediately some of this label enters the fine fractions, and specifically the clay fraction here, the 13C goes through and declines a little bit already after 42 days, whereas the nitrogen label enters the microbial biomass and stays in the soil, whereas some of the 13C with time is then released again as a 13CO2. But most of this enters the very fine fraction, the clay fraction, smaller than 2 micrometers. When we then look at these fine fraction, we see, first of all, I hope you can see that this is scanning electron microscopy that we always do before we go to the NanoSIMS, because otherwise the NanoSIMS resolution is so high that we are lost in our sample. So we first have to get an overview, and we do this with the scanning electron microscopy. And we find a lot of clay particles. We find, first of all, that only less than 26% are single mineral particles, and a lot of them are mineral clusters. So we have single particles like these here, but a lot of them are clustered, although we tried to disperse the soil and to release only primary single minerals. So and this is also something that we have found in soils. It's very difficult to produce these single minerals without disturbing the organic matter completely. So we have a lot of this organic matter here, and this is also what we then see when we look at the 16O signal in the NanoSIMS. We see these surfaces quite well. If we then look at the label, this is the same SEM, and here in yellow we have the 12C carbon and the 12C carbon and nitrogen, and we see that they are very heterogeneously distributed. It is not homogeneously distributed, and this changed a little bit the ideas how soils accumulate carbon, because there was a theory of a kind of monolayers that are homogeneously attached to the surfaces. What we see is that it's more in some spots, and when we do much more of these observations and make an estimation, we find that around 20% of these surfaces are covered at all. And this is the 12C and 14N, so this is the old carbon in the soil. Again, this is the old carbon here, and then if we look at the 13C and the 15N, so the green spots and the pink spots, you see that, again, this is very heterogeneously and distributed and found only in specific spots. And what we also see is that these newly formed organic matter is in the same organomineral clusters that already had old carbon present. So the new carbon is not distributed homogeneously, but it is formed only at specific spots. And the 15N, if we also find 15N, this is always because the 15 or the nitrogen generally goes through the microbial biomass, this is always a hint that the microbial biomass is really relevant here and is part of this accumulation. But of course, we made a lot of sample preparation and we disturbed the initial structure, but still we see that we have these mineral surfaces that remain partly free and are only partly covered. And this has implications for carbon sequestration in soils, but also for other soil functions, because this means we have these small spots where we find this new carbon, and we have these spots where we have generally carbon in the soil, but a lot of the surfaces are available, and at these surfaces, other things can happen. These surfaces can produce cation exchange. They can be, if they are iron oxides, they can participate in redox reactions. In general, these surfaces, of course, can also participate in weathering. So we then considered that we should look more closely how this was a lab experiment of 42 days, how this works in nature. So we looked at the development of microaggregates after glacier retreat. This is the Dammer Glacier in Switzerland, and yeah, it was around here 140 years ago, and since then the Dammer Glacier has retreated, so the glacier foreland is now here, or was here at around 2010 when we sampled this. And when we go down, we have a chronosequence of soils that are very young, 15 years old, 60 to 70 years old, and 110 to 140 years old. It's difficult because it's very heterogeneous to have better values for the age of the soil. But generally, we see a nice soil development. And we also looked at mature soils, which are outside the glacier foreland generally, and they are at least more than 700 years old. Probably they are much older. We also, again, looked at the fine particle fraction, first with scanning electron microscope. Here it's explained that we apply these then in a suspension on a silicon wafer, and we get these samples then, and yeah, we do NanoSIMS again, and we get these elements. You now know this already. Here we have no labels, so we only look at the natural carbon-12 and the natural CN. And yeah, and what we did here, we made a lot of these measurements with many parts. So oxygen for the surface, carbon for the organic matter that does not have nitrogen, and CN for the organic matter that also has nitrogen. And we then get a lot of these observations, and so first of all, we try to separate then the mineral surfaces and the organic matter coatings in our NanoSIMS observations, and we calculate the degree of SOM, of soil organic matter coverage, and also the connectivity of this organic matter. So if they are single spots like here, or if they are strongly connected. And when we've done all this, we can look at these data. And first of all, what we see is two fractions. Both of them are generally heavy fractions, but the lighter one, this is 1.6 to 2.2 grams per cubic centimeter. This is the fraction that contains a lot of organic matter, because it makes the minerals, the organic matter generally makes the minerals lighter. The heavy fraction, which is heavier than 2.2, contains very little organic matter. And with time, we see that this heavy fraction is only a small proportion, and in the mature soil, it's almost non-existent and has very little SOC concentration, and this lighter fraction increases. So we see accumulation of organic matter in this fraction, and this is what I wanted to point out. This heavy fraction, with time of soil development, there's a little bit heterogeneity in the data you see, because this glacier forefield is really, the soils are quite heterogeneous soils, and so we have some scatter in the data. But you see that the mature soil has almost none of this heavy fraction, and this means with time, all the mineral surfaces take up organic matter as soon as they are provided by weathering. So one thing that was something I learned in the last years is that it's not the accumulation of organic matter and the formation of organic matter that is the limiting step to produce well-structured, organic-rich topsoils. It's most probably the amount of mineral surfaces that are present and the rate of weathering that produces them. If we have these weathered minerals, they immediately associate in the processes that I've tried to describe before in organomineral associations. And again here we see that the coverage of organic matter with time increases and specifically in the very fine the materials that are still finely associated organomineral associations, and also in the mature soil. But what we also see that even after a long time of soil formations, these mineral surfaces are only partly covered. In this case, I think it's something like 59% or so in the maximum. And we also see, if we look at the connectivity of the organic matter, that this increases with time and specifically in the mature soil. And this confirms what we've seen in our lab experiment, that the growth progresses from already existing organic matter patches, but they, if time is longer, they enlarge and produce larger associated particles that have a lot of organic matter that is connected to each other on these very fine particles. Small, very small microaggregates, let's say. So how does this growth most probably work? We think it really is a process that is associated with the growth of the microbial community in the soils. And this is an incubation experiment that Peter Kopitke from Queensland University did, and looked also at the data with us with NanoSIMS. And what we see specifically is that nitrogen-rich compounds accumulate. And we think that this is an indication that it is microbial products. And these new associations also go on beyond these spots and attach with organic matter-free mineral surfaces. And this is where we find specifically these nitrogen-rich compounds. And so we think it's very important how the microbial habitat is and how these microbial communities can grow in soils so that they produce these organomineral associations via their exudates, via their necromass, and that this still needs that three compounds need to come together at a spot of at micrometer scale. And this is, we need a mineral surface, we need organic matter that is decomposable for microbes, and the microbe must also be at the same spot to produce such organomineral associations and to make them grow. And the microbes are at these spots where there was organic matter before, because this is how they lived there. And then they start to sleep for weeks or months or years, because the soil for the microbes at some spots is like a desert. So they wait for new substrate to come. If new substrate comes in, either by bioturbation, because an earthworm brings in organic matter, or because we plow the soil, or because it's dissolved organic matter that is leached in the soil, then the microbes start to decompose that, and this is at the same spots where organic matter was formed previously. So the role of microbes and their habitat that they form themselves is very important in the exploration and covering of these mineral surfaces. So my last example is one aggregate. What I showed until now was mineral particles that we dispersed and isolated from the soil. And even there we found we have very fine microaggregates still remaining. But this is again this soil in the Cheyenne Research Station where also the incubation experiment was done with that I showed before. This is a Cambisol Braunerde in German, north of Munich. It's tertiary sediments covered with loess, and it's arable plots under long-term crop rotation. And the soil looks like this. This is the plowed topsoil, and this is where we took the sample from. And in this case, we did not disperse the soil, but we isolated aggregates. These are the aggregates. This is 1,000 micrometers. So this is larger aggregates that we isolated from this soil. We embedded them in resin and cut and polished the surface. And as I have mentioned, polishing is very important for NanoSIMS because otherwise the data are not very good and the resolution goes down. And then we always do light microscopy, scanning electron microscopy, and we look at the element composition. And we also did NanoSIMS, specifically NanoSIMS analysis with this aggregate here. And we made a regular grid. This is the yellow spots that you see here of 42 spots. The spots are not to scale, of course, but we covered with these 42 spots 1.2% of the polished surface of this aggregate. And we analyzed it for carbon, oxygen, nitrogen, Si, aluminum, and iron. So organic matter and minerals like we always do. But in this case, it's one aggregate. And to be honest, we had really problems to publish that because the reviewers said, you talk about one aggregate. And we didn't do 20 aggregates. But we have a very nice coverage of the distribution I will show of these elements in this one aggregate. And we have a much better coverage than what we do if we work on the plot scale in the field and we sample one soil with 3 replicates. So it's really a very detailed study, but yeah, was difficult to convince other soil scientists, but finally it worked out. And we had, with all these data, 11 million something points measured. And we then looked at these distributions of nitrogen, of Si, for example, of aluminum, and there was a lot of spectral smoothing and normalization of these data and a lot of multivariate statistics. And this is what we did with Derek Rogge from DLR. And we used an approach that is normally used in remote sensing and applied it to look at our aggregate. And what we did is we then made, for each individual point that we measured, the distribution of all the elements together. And yeah, we tried to see if this is random or if we get a clustering. And finally, it turns out we get real nice clustering. First of all, we get what we did here in gray. This is the pores. They have only carbon, and this is because this is the resin, nothing else. And we have nitrogen-rich organic matter, which is particulate plant residues, the leaf or root residues partly decomposed. We find aluminum-rich mineral grains, Si-rich mineral grains that have Si and mainly oxygen. And we find finally parts where we have aluminum-rich clays with some organic matter and Si-rich clay minerals. And if we try to combine all this, what we get is these patterns. And what was new for us and what I think is interesting to see, in this one aggregate, we have two types of microdomains. It is not pores and mineral grains and clay minerals are not randomly distributed, but we find these structures here, and we find these structures here, what we call microdomain C and microdomain A, and B is where they meet, basically. So what you see is that these materials here, they have a lot of large gray parts, so they are, I can't let that go by, they are pretty porous. So, but these materials are not. And if we make calculations, and they are composed of two different types of materials. Basically, these large blue minerals are quartz or feldspar grains. When we do this in detail, we see that these Si-rich mineral grains are almost exclusively in the A domains, and the aluminum-rich mineral grains are in C. And yeah, Si-rich clays are in A, and aluminum-rich clays are in C. And yeah, the red is iron oxides. Unfortunately, the iron oxides are only detected if they are large iron oxides, because we are at the limit of resolution with NanoSIMS for the small iron oxides. But also the pores, you see here, the pore area is much larger in A, and the particulate organic matter is almost exclusively here. So this is most probably these particulate organic materials where the microbes grow around and consume these plant residues and mineralize a lot of them, but also fill the pores and make them, by growing, make them much smaller and probably inaccessible. And we also calculated the number of pores, which is different between A and C, and the median of the single pore area is higher in A than in C, and the maximum single pore diameter is much higher here in A and compared to C. And if we summarize this, we see that the microdomains here are quite different, and we have here large pores, as we have seen before, around large, most probably feldspar or quartz grains. There is thin layers around with phyllosilicate clay minerals. There is some organic matter here, but it's low. And we have many large pores and a high total pore area, whereas this microdomain has the blue ones are small aluminum-rich primary minerals, probably feldspars. This is the blue is quartz here. But it's in a phyllosilicate matrix that is most probably illite, which is the yellow ones. And it is, these are high, these yellow ones, and these phyllosilicate minerals are rich in organic matter. And they surround these green residues of particulate organic matter that remains in soil, and we know that such particles can remain in soils for 10, 20, 30, 50 years by radiocarbon analysis and other isotopic approaches. And we think this is mainly because the pores are small and narrow and few, so that they are just not accessible anymore for the microbes. And so in this microdomain, we have a sorptive potential also from the clay minerals that is accessible because water and solutes can enter via the larger pores and also be transported. These pores have a size that is habitable for the microbes, and the organic matter that is surrounding here is rather easily accessible. We know that if the organic matter is sorbed to the mineral surfaces, microbial degradation is limited, but we are not sure how much this is exchangeable. Most probably some of this organic matter is exchangeable. In this microdomain here, and remember, this is in one aggregate of millimeter size. We have a sorptive potential, but this is not available because there's not many pores there. And most of these pores are not available for microbes because they are too small, which also means water transport is, of course, you know that, much slower here. And we think because the microbes can't live very well here, the organic matter is quite protected against decomposition here. And this means this is the areas where organic matter accumulates for a long time in soils, is stored, and if organic matter via CO2 binding through the plant biomass enters the soil, here it stays probably for a long time. And this also means the CO2 that is taken from the atmosphere can remain in the soil as organic matter for hundreds or probably even thousands of years. So in summary, we see that this microstructural arrangement of mineral platelets, clay minerals, larger, even small but larger quartz or feldspars, and iron oxides. This is should be iron oxides. We think it's like a house of cards structure that is partly filled with this organic matter. It can become filled more if we add organic matter to the soil. It defines the microbial habitat, allows organic matter accumulation in the soil, but this is independent from the pure specific mineral surface area, because the accumulation does not occur, as we thought previously, just on the surfaces, but in the three-dimensional structure that is made by the minerals in association with the organic matter. But this also means that the organic matter, the capacity of soils to store organic matter is not limited by the mineral surface area, but can go beyond. And of course, if we do not feed the soil all the time with organic matter, we lose this organic matter again. To conclude, we think that the observations of soils and their composition at the scale that I tried to show here. This is micrometer range, and below is specifically relevant for understanding the emergent soil functional properties that derive from this structural arrangement. And we see that soils have a high potential to sequester organic carbon and at the same time fulfill other ecosystem services such as storage of nutrients via cation exchange capacity at the mineral surfaces, or binding of pollutants to the mineral surfaces, and so on. And we think we need to go more into detail in soil science to understand this intrinsic capability of the clay-sized mineral mixtures. And what we looked at is just mineral mixtures from Bavaria. So there's many other soils in the world that have a much different and very specific composition of minerals that is really different to what we looked at. So we need to look at this capability of these clay-sized mineral mixtures in soils to form these emergent soil aggregates and the dynamic changes in the pore structures that are induced by, of course, physical processes, but also the microbial processing of organic matter. And we think, therefore, that soil type and mineral composition matter because they define the microstructural arrangement of organic matter and minerals, and at the same time also the microbial habitat, which vice versa then is responsible for formation of the soil structure together with physical processes, of course, and yeah, their own habitat. I thank you for your attention, and of course I have to acknowledge the German Research Foundation because they funded the NanoSIMS instrument and the priority program biogeochemical interfaces in soils, as well as the research unit MAD-SOIL, which means microaggregate formation and turnover in soils. Thank you very much.
Thanks a lot, Ingrid. Thanks a lot. I think we have time for questions. I'm really impressed when I'm in my garden at the weekend. I'm sure that I have another view to the soil. Questions. I think we have time for questions. Can you raise your hand that I can see who's willing to ask the first question? Cynthia, please.
So you mentioned that it is
Can you stay up a little bit, then the other ones can see you in the...
you mentioned that it's very hard to say your findings are only, well, about the Bavarian area, about the German soil structures.
Mm.
Can you somehow extrapolate what you expect on other soil structures?
Of course, this what we find is, we think, typical for a lot of loess-derived and cambisol type temperate soils. So the same you would also find in prairie soils in the US, for example. So in the temperate zone, I think a lot of that can be extrapolated. But of course, if you go to tropical soils, where there's very little clay minerals remaining because they are much more weathered and you have much more iron oxides, the structure is most probably different because the iron oxides are much more dominant compared to clay minerals like here. And of course, there's other soils where we just start to explore what happens, like permafrost soils. When they now, the organic matter that's there, it's frozen, but when they start to melt, it becomes important if such minerals are there and if they can form these organomineral interactions, which would be good, because then at least some of the carbon will remain in the soil. Otherwise, the permafrost carbon will be released to the atmosphere, and that's a real large pool.
Thank you.
Questions, please? Yeah?
I would have a question, a technical one to NanoSIMS. These pictures by the electromagnetic field.
Can you use the microphone, please? Then it's better for the YouTube.
Technical question to NanoSIMS. If you make a picture using electromagnetic fields separating the mass units, they are picture as a whole, or is it like a raster?
It's like a raster.
It's raster.
I mean, it's rasters,
Yes.
yeah.
Okay.
What we did not do very much, or I haven't shown, is you can also stay at one spot and just drill a hole inside. So you could also see
Like
depth
sputtering.
distribution of minerals or of any materials that you drill in. But we sputter off and try to sputter off the same amount of organic matter or surface all the time. Of course, when you now see how complex the interaction is with the minerals and the organic matter, the sputtering off is not always to the same depth, so it becomes complex then how this looks like. And we have also looked at the surface of our samples after we measured NanoSIMS with NanoSIMS, and then the surface gets like hills and valleys.
Okay, thank you.
Additional questions? Ingrid, I have two questions. The first one, you know, I'm a modeler, a stupid modeler, and modelers, they like to integrate different kind of scales. And you have mentioned scales from nanometer to 63 centimeter. I hope I got it right. How do you bridge the scales? How do you get the scales together if you analyze how much CO2 you can store in the soil?
If we want to look at really the carbon storage, we don't go into these scales.
Okay.
Then you do the same raster analysis over a whole plot.
Okay.
And then you, you depends on also the heterogeneity of the site, but this is a completely different scale.
Okay.
But we have quite nice carbon maps for Bavaria, because Bavaria 20 years ago, no, almost 30 years ago, decided to make a raster sampling over the whole Bavaria, distance 8 kilometers, and then we have 1,500 soil profiles, and then you can do this at this scale. And now there's very nice data from an inventory from the Thünen Institute in Braunschweig. They did all Germany, only arable, and because they Ressortforschung for agriculture, so they did only agricultural use soils. But we have really very good data now on the carbon storage in Germany. This just published last year.
Now I have an
But this was from Thünen Institute.
Okay. Now I have an unfair question. You know, I think I'm 100% sure that you get this question 100 times before. How much CO2 you can store related to the climate change? You know, this storage behavior must be strongly dependent on the climate, with other words, on temperatures and so on.
Mm-hmm.
And is there hope, if we get an increase of temperature, that we get more storage capacity? Or is it completely different to this, if you think about permafrost areas?
Permafrost is a difficult problem, because this very much depends how this thermocast develops, and there's only few studies that really went into soils in Siberia and Alaska and so on. But I'm not sure how much we can
store, we can cover, and
we can stop from being released, because this is just, I mean, it's stored since the ice ages, and now it melts, and this is difficult. With other soils, there is the 4 per mil initiative that says we can compensate the increase in the next 20 years if we store in all soils globally to 30 to 40 centimeters if we increase the storage by 4 per mil. The major problem is some soils, our soils are not really depleted. So we will, in temperate regions, we will not really be able to store 4 per mil every year additionally. I think this potential is specifically there in areas where the soils are really depleted. And so for Bavaria, we have recently published an estimate of 1 per mil. But even 1 per mil would be great.
I think it's really
because
huge
it's a huge amount. And if we then go to soils that are depleted or where the input can be increased, because as I tried to mention, you have to feed always carbon, organic matter in the soil. So you need this organic matter. You have to produce it. And you have to additionally, and this is why this is limited in Germany. We already do a lot of crops over winter and Zwischenfrucht and all these things. There can still be done something, but we think the potential in Bavaria and probably also in Germany, and there's similar results, I think, from Switzerland, is around 1 per mil. But where you can increase yields, which means if more biomass grows on the soils, then you can also, then you always have more input. You have more roots, you have more root exudates, you have more crop residues, but this means you need to increase yields. And this is a lot of these soils are in the global south, and there's more economic constraints and things like that rather than soil constraints. And then, of course, plant growth is limited in some regions by temperature, by dryness, and so on. So, for example, in Australia, they have carbon certificates now for soils. This is what also will come up here, I guess. But they lack the organic matter because a lot of the large parts of the country are just too dry to produce organic matter. And you can store it, but then you have to produce it.
Okay, wow. Okay, yeah. There's a question from the YouTube. Simon, can I offer the microphone for you?
So the question is concerning the pores. Does organic C decrease pore connectivity by blocking pore necks, or does it create new ones?
I think I don't know many data on that. I would think that if you are at a specific spot where there is an organic residue that is decomposed, it will block with time, clog the pores. Of course, what we look at is only part of the whole story. So some of these clay minerals, they swell and shrink and have a lot of physical processes they undergo. And so in the long run, the soil as a whole will be more porous. This is well known. It will be more porous if you add organic matter, and the structure will be generally with much more and also larger pores. But I think this is then in the whole. What we do not understand, or I do not understand, is we always look at formation of the whole things and the aggregates. But we know the aggregates have a turnover. And we don't know how this happens, how they break apart so that the organic matter becomes accessible again and they rearrange. We know they do that from turnover studies that has been done quite often, but the process is not as well understood.
And how long is this time scale for this kind of rearrangement?
Macropores, some months.
Oh.
Macro aggregates, some months. Micro aggregates, 250 days or so.
Okay. Is there
But that's temperate soils around.
Simon, second question.
Okay, the second question is a more technical one, and it's, can you image also MN too? So
yeah, I think so.
Okay.
I mean, the instrument we have is limited to 7 elements or isotopes at the same time. But if the spot where the MN is large enough so that the resolution with the 50 nanometers is met, of course, you can also image in soils MN oxides or things like that. You just don't look then at aluminum or iron and take something else. You can do almost, I mean, there's some restrictions, but many elements and 7 at the same time.
Simon, there isn't any kind of additional question? Super. Ingrid, thank you very much for this very impressive presentation. I learned a lot personally, and I hope you have learned a lot too. And it's an honor and pleasure to have you here, both of you. Ingrid, you and Peter. Peter, you know, is our Aggregate lecturers in 2018, correct, Peter? And I hope we will see us again as soon as possible, and that you get a good feeling. Please, you can share it with yourself, but you have a chance to share it with Peter.
Yes, of course.
Thanks a lot. Thank you
very much. Thank you very much.
Thanks a lot.
Thank you for your attention.
And thank you much for the YouTube users and for the nice questions that they have formulated. And I hope we will see us again at 6 o'clock. Then we try to we will opening our exhibition, our Porous Media exhibition. And I hope that we can tell you a little bit more against what Ingrid has mentioned about pores, porous media, and flow and transport and deformation processes in porous media. See you again in half an hour. Thanks a lot.